I think you know that the A level, AQA International Biology, it has 2 units for AS and 3 units for the A level, 1 and 2 AS, 3, 4, 5, the synoptic paper related to the practical in A level on, right? Let's simply كده say quickly some things about the board and the assessment of that board. We said that we have 2 units, unit 1 and unit 2 AS, 3, 4, 5, all A level of A2. Okay, what are the content of each unit or specification will be available to you in any site, it's there. Okay. As a whole, the content of the AQA, I feel it's simpler than Cambridge and Edexcel. I am teaching the 3 boards long ago. Oxford is simpler, and some details may be not needed as much as it's found in Cambridge as well as Edexcel. Okay. The questions also have the style of simple, but they need good writing. And you must have writing skills. As a whole, in biology in general, you must have writing skills. Yes, there is no deduction for the spelling mistake, for the grammatical mistake, but spelling mistake, grammatical mistake can change the meaning of the sentence or whatever you want to say, so you will not take the mark, right? In Oxford, especially, we have to pay attention how we are writing the answer, which word we are selecting to write our answer. And this will easy to be managed by solving paper as much as you can. You will say Oxford don't have that much resources. No, Oxford, AQA is very old. It's only introduced for the international. We start to be common for us in recent years, but there are papers from 2013, 2012. We can select from these papers whatever we need according to our specification, and we will use. You got it? The uniform mark, it's 100, and this will be taken as over 100. Okay, and then all will be added together. The uniform mark is 500 for A level. You must achieve 270 marks from this 500 from A2 to get the star. I will talk about that later. If anyone's taken want to complete A2, I can explain in detail how the caching would be calculated. Okay, this is the code 696119612 for AS and A2. Let's go to the explanation. But before going to the explanation for the student who will start to attend the course with me, okay, I would like to explain what we are doing and what is the plan of our course, right? During the classes, we have explanation. I will explain for you whatever you need, whatever you like to understand. I will give it to you with notes, with everything. Any material you need, from material to study, classified questions, it will be there. After each session, I'm explaining, I'm sending questions to solve at the homework. Clear? After each session, during the session, you have to be sure that you understood everything. I would like the camera to be opened because it's better. It will make more contact between me and you, right? Sometimes when I'm looking at your faces, I can't see if you understand or not. You want me to repeat, you have doubt in a point, even without talking to me, I can see that, and which make it easier for me, okay, as a guideline for me to finish the lesson smoothly without being fear or doubt that you did not get any point. If you don't understand any point, feel free to ask me, right? You have to ask and discuss and even argue with me any point that you didn't understand or you did not get it during the session. After the session, you have to study whatever I gave to you. Okay, if you have doubt, you have problem, you did not understand that part, when you start to read it alone, call me, send me message, right? Send to my assistant message. Miss, I have problem in that part. I understood it during the session, but now I did not It's not clear for me. Could you repeat it for me one more time? Could you help me in that? I will be ready to do that, but before the next session. Don't wait till we will start next session and you will start to ask me, Miss, I did not understand last session. This is wasting the time of the session, right? Because at the beginning of the next session, whatever we cover today, I will recap it. I will ask you, give you questions, whatever my own question or from the past paper, but I have to follow it up. I have to follow you up. I have to be sure that you are studying, you are aligned with our plan, right? Don't delay studying for any time later. I will study when the whole chapter finishes. I hate this. Day by day, you have to study. Don't be behind the others. Because after that chapter, we have new chapter. After that unit, we have new unit. So when you delay your work, you will never catch up the others. And we have time plan. We have to follow that. The difference between good student or good teacher or in any job, the planning. You have to plan for yourself when and how you will finish your missions. You understand me? We have Google Classroom for the assignment, for the homework, for the material. Okay, if you assign your work on time, it's fine. So it will be checked. You can discuss your doubts. It's okay. But if you assigned it after the time of the dead time of submission, in that case, who will help you? Who will check the paper for you? Or you are working in another part, working another homework. So you will be the loser because no one will come back. We want to go ahead. So finishing your work in time, okay, it makes it better for your study and me to follow you up. You got my point? The following up will be by me from my side, me and my assistants, and I will contact your parents with a separate group, right? Also to send the reports, to give them the assessments, or if there are any exam paper, I would like them to print and give it to you under their supervision. As sometimes also you will attend the test with me face-to-face or online. If it's available for some student to come face-to-face, it's fine. This for our system, right? Before the examination, we'll have classes till the day of the exam. Okay, after finishing the syllabus or the contents that we have to finish, schedule of revision will be given to you, what exactly you have to do daily till the day of the exam. Let's go with the explanation, because already if you will go to Jan, you have to start because the time is limited. Okay, I will start with Unit 1. Unit 1 starts with easy topics like biochemistry, which related to biological molecules and cell structures. Yani, most of the basic things that you have to learn in biology before going to Unit 2 and Unit 3. Unit 1 starts with biochemistry, or are talking about the biological, okay, molecule. Okay, what are biological molecules? Anyone of you has idea about the biological molecules? Biological molecules, biology mean living things. It means the molecules which found inside the body of living things. Actually, if we will make analysis for our body, at the end, we made up of few elements only: carbon, hydrogen, oxygen, nitrogen, may sulfur, may some electrolytes, right? At the end, we compounds interacting together form different structure which could make chemical reaction, or structures which make our body, like bones, muscles, hairs, nails, tendons. The biological molecules which found in our body, right, they categorize into 4 major groups, right? Okay, and if you had IGCSE before, you must hear about carbohydrates, right? Or proteins, lipids, as well as DNA or genetic information. All living things must have these 4 biological molecules. Okay? That chapter, we will learn about the chemistry of these molecules, right? Before that, we learned carbohydrate could be glucose, sucrose, starch, proteins like enzymes, antibodies, lipids, oil, solid lipids like butter, DNA, RNA. Okay, but this time in A level or AS level, we should learn about their structure and how their structure correlated to their function. Okay, what is the main component of carbohydrate and what is the function of each part? Okay, let's start with the carbohydrate. For carbohydrates, from its name, carbon, okay, carbo, it's related to carbon. The word hydrates usually related to water. So why is it called carbohydrate? Because if I will go to their empirical formula, if you know what is empirical formula, do you hear it about empirical formula? Huh? In chemistry, if you had it before, or the simplest whole number ratio of carbohydrate is CH2. This means carbohydrates made up of carbon and hydrogen and oxygen, okay? And the ratio between these elements found exactly as if carbon added to water molecule. 1 carbon, 2 hydrogen, and 1 oxygen. This is the ratio between the carbon and hydrogen and oxygen if they are found in the carbohydrate molecules. Is it clear? Okay. Right. We learned about carbohydrates that they could be made up of 1 sugar called monosaccharide, disaccharide, polysaccharide, right? Okay, so we can divide the carbohydrates, according to the number of the sugars which found in 1 molecule, monosaccharide, disaccharide, polysaccharide. Okay, when I say mono or di or poly, you should know what is the meaning of this term. Mono means 1, di means 2, poly means more than 10 or than 10, or usually we call it large or macro structures. Monosaccharide, I think saccharide coming from the Arabic word sukkar, if you know it. Saccharides, okay, disaccharides, and polysaccharides. Okay, monosaccharide means the carbohydrate, okay, made of 1 sugar. And examples of these monosaccharide, the most famous one, glucose, and its isomer, which is called fructose, and we'll learn them why they are isomer to each other. And there is another one called galactose. Okay. Disaccharide, okay, like sucrose, which made up of 2 sugar. Disaccharide made of 2 sugars. Sucrose, and I think you're familiar with lactose, maltose, okay? Sucrose made of glucose plus fructose. Lactose made up of glucose plus galactose. Maltose made up of 2 glucose molecules, okay? We will handle each one in detail. And about polysaccharides, okay, polysaccharides, okay, they made up of more than 10 sugar, 20 polysaccharides, which is starch. We have glycogen in animal, and we have cellulose. Okay, when I say the word polysaccharide, okay, it reminds me by word called polymer. Did you hear about polymers before? Okay. Huh? Are you familiar with that word which is called polymer? Polymer, it is large, okay, molecule. and we call it macromolecule, made up of small units bonded to each other in that way. And these small points called monomers. Yani, if I have some beads, okay, you know, some girls may used to have that when they were young. They have beads and they can connect these beads together to make bracelet, to make necklace, and this one. This as a whole will be polymer. And each single bead called monomer. As I said now, monomer means one, poly means many. So the polymer, it is large molecule made up of smaller units called monomers. You got it? These are the different carbohydrates if they have one or two or more than sugar bonded to each other in the same molecule. Okay, let's go to see the monosaccharides. Okay, monosaccharides, okay, they are carbohydrates which are made up of one sugar. طيب, let's define the structure or the chemical structure of sugars first. I think most of you handled organic before, okay? If I say they are hydrocarbons, they are organic compounds which are made up of carbon and hydrogen that bonded to many hydroxyl groups. The compound which related to carbohydrate is hydrocarbon chain but has many hydroxyl groups. Okay, I will give you example. Okay, like glucose. I will just show you the glucose structure if it's long open chain. Glucose, one of the carbohydrate which have that formula C6H12O6. We learned that in respiration when we had aerobic anaerobic respiration in IGCSE level, right? Okay, so at least I know that glucose have 6 carbons bonded to each other. 1, 2, 3, 4, 5, 6. And the first carbon here has functional group, or this functional group, this part called aldehyde. And after that, each single carbon found in that molecule bonded to one hydroxyl group. Hydroxyl group has that formula. So here, there is hydroxyl group. Here bonded to hydroxyl group. This hydroxyl group. Each carbon bonded to one hydroxyl group. طبعا, plus the hydrogen to complete the 4 bonds of the carbon. This is the structure of glucose? Yes. This is structure called aliphatic structure or the straight chain of glucose. Do you understand why they have many hydroxyl compounds? Because they bonded, each carbon bonded to one hydroxyl group. The glucose has, okay, general name called, can you understand the meaning of that name? Yes. Hex means 6 carbon. O's, this related to sugar. If you notice, all of the sugar ends up by suffix O's. Glucose, sucrose, pentose, ribose, galactose, right? So when I say hexose, it means carbon have 6, saccharide have 6 carbon. When I say pentose, saccharide has 5 carbons. Is it clear? Tamam. This is the aliphatic structure of glucose, but usually we are not handling that structure of glucose. Okay? Usually we are using the ring structure of glucose, which is easier, right, to use, okay, and handle it if you talk about the reactions of glucose and all of that. Okay, how can I draw the ring structure of glucose? Simply, you can see this is the aliphatic or straight chain of glucose. Imagine it is a snake, and that snake will bite its tail like that. So this is carbon number 1. Okay, this is carbon number 1, and it will go and react by that oxygen with carbon number 5. Carbon number 6, no, not involved in the ring formation. So how the ring structure of glucose could be? Okay, it is hexagonal ring. So I will write about the ring structure of glucose. It's hexagonal ring, yani ring has 6 sides. Okay, and that oxygen will be a part of that ring, yani it's in the ring. Because simply imagine with me, this bond will be removed and it will be made between the oxygen and carbon number 5. That's why the oxygen, okay, will be in the ring. So how can I draw the ring structure of glucose? I will draw the oxygen, then I will complete the hexagonal ring in that way. What about the hydroxyl group? How I will arrange them? Where is carbon number 1? It's clear. Carbon number 1 is here. And I said the bonding will be between carbon number 1 and 5, carbon number 1 and 5. So 1, 2, 3, 4, 5. And here, carbon number 6 out of the ring, as I told you, with its hydroxyl group. Is it clear? 5. Carbon number 1, the hydroxyl group will be drawn down here, and the hydrogen is supposed to be here. Carbon number 2, the same, hydroxyl group will be down. Carbon number 3, it is the only one which found in the other side, as well as number 4 will be down like the others. Okay, so this is the ring structure of the glucose. Okay, let me draw it one more time. So again, I will make hexagonal ring at which the oxygen is a part of the ring. Is it hard? No. There is carbon number 1, carbon number 2, number 3, number 4, number 5. At number 5, I will put here CH2OH. What about carbon number 3? It's only different side than the other. Carbon number 1 down, carbon number 2 down, carbon number 4 down. Okay, this is for the structure of glucose. Okay. Anyone can ask me, what if I will draw the hydroxyl group in different direction, especially the hydroxyl group in carbon number 1? And look at that hydroxyl group in carbon number 1. What if I will draw that hydroxyl group up and the other one I will not change because they are not in the functional group? We said the functional group is that group. So it could change its position, right? Okay. What if I will draw the hydroxyl group in carbon number 1, okay, to be above the level of the ring? By the way, this position, it's below the level of the ring. You understand what does it mean below the level of the ring? And this one is above the level of the ring. Below the level of the ring, or above the level of the ring, this related to stereochemistry, or we have to look at the compound in organic chemistry, not only to their structure, as well as we have to learn about their geometrical arrangement or how they looks like as 3D shape structure. To make it easier for you to imagine with me, the glucose ring is the table on the surface of the table like that. Here is the ring of the glucose. When I draw this one hydroxyl group down, it means this coming below the level of the ring. So this one is below the level of the ring. Carbon number 3, no, it is above the level of the ring, going up the ring. Carbon number 4 going down. Okay, you got it? Do you imagine with me what do I mean? And this one also to the level of the ring. So here is the level at which the ring of glucose is found, and here is the hydroxyl group. If it's found below the level of the ring in that way, that glucose is called alpha glucose. That glucose will be called alpha if it will be arranged below the level of the ring in that way. And that one, if it alpha, the opposite of alpha is beta, right? Not opposite, يعني always if we classify things as alpha, the other one will be beta. Beta, it means the hydroxyl group of carbon number 1, only I'm looking to carbon number 1 only, above the level of the ring. How? It could be the same. Here is the table, right? Okay. Here is the glucose ring on the table, plan the table. Carbon number 1, its hydroxyl group up with the hydroxyl of carbon number 3, and all of the other stay below as it was. Okay, so here I learned that the ring structure of glucose can have 2 stereoisomers or different arrangement in the geometry. It could be beta glucose or alpha glucose. What is the difference between them? In alpha, I will look at carbon number 1, the hydroxyl group below, or you can see it's far from the oxygen of the ring. Beta glucose, no, it's above or close to the oxygen of the ring. So OO like BB هنا. You got it? You understand the difference between alpha glucose and beta glucose? Okay, I will say it one more time. First of all, we are talking about the structure of glucose as one example.
Miss, can you give the alpha and beta structure?
Yes, I will repeat now. Okay, I'm repeating now from the beginning. It's fine. I will talk about the monosaccharides. As an example, we will study the chemistry of the monosaccharide. Chemistry means I have to learn about the structure. 3 years we are studying that glucose is C6H12O6, but we never learn how this carbon and hydrogen and oxygen bonded together. Okay, first of all, it's a hydrocarbon chain which has many hydroxyl group bonded to each other. And if I will draw them as a straight chain, it will be like that, right? 6 carbon, 6 carbon bonded to each other. Okay, 1, 2, 3, 4, 5, 6, each one bonded to hydroxyl group, as you see. And 1 carbon, which is carbon number 1, has functional group called aldehyde. Why it's called functional group? Because if it will react, it will depend on the presence of that group, because it's a characteristic for the whole molecule. Okay, glucose can be found as aliphatic structure or straight chain like that. This is called the straight chain, or ring structure. What do you mean by ring? It means cycle. Okay, and the cycle in organic compound have to be formed by reaction of the 2 carbon which found in the long chain molecule. That reaction happened between carbon number 1, carbon number 5, okay, carbon 6, it's out of the ring. So if this oxygen reacts with that carbon, the ring will be closed. You got it? Okay. How the ring will be formed, how the ring will be closed is not important for me. I just telling you to understand. For me, I would like to understand only how to draw the glucose as a ring structure. Okay? Right. We said it's a hexagonal ring. So I will draw the ring, but inside this ring, there is oxygen as one of the angles of this ring. Is it clear? Is that part is okay? Okay.
Yes.
Right. Then we have to learn to count the number of the ring, right? Carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, but we know that glucose is 6 carbon or hexa, so H2O, OH is here, because I told you this one not involved in the ring formation, it's out of the ring. Right, okay. Carbon number 1 and carbon number 2 and carbon number 3, carbon number 4, as I said, each carbon in that molecule must be bonded to hydroxyl group. Is it clear? The hydroxyl group of carbon number 1 and 2 and 4 are down. When I say down here, it means below the level of the ring, like that table. You understand this 3D shape structure? You know that? You got that point or not? You got that point?
Yes.
Okay. So when I'm saying down, it means below the level of the ring. Okay. But carbon number 3, no, all the time it is the opposite. It's above the level of the ring. Even when I draw it here, it is the only one which is up and the other all below. Okay. But this is the ring structure of glucose, right? But after that, they found it. If we look at carbon number 1, the hydroxyl group could be down like that, or could be above the level of the ring like that. Okay, so how can we differentiate between them? Okay, so we will call it alpha glucose and this one beta glucose. Okay, what is the difference between alpha and beta one more time? They have the same structure, same everything, but the stereo structure of the molecule, the OH could be, while the formation of the glucose, right? The OH was below the level of the ring, so in that case it will be alpha. The OH will be above the level of the ring, in that case it will be beta glucose. You got it? You can annotate on the board. I will allow annotation and I open annotation for you. If anyone, if you can draw alpha glucose in the board, okay, can you? Type, I will draw the structure and you will tell me it is alpha or beta, okay? I will draw the structure and you will tell me if it's alpha or beta on chat, please, private, okay? Okay, good, Islam. Islam is very fast. Hamza also. Who else? Muhammad, good. Who else? Isra, you ask it. Still, you don't know up till now? Islam, Saif, Tasneem. Yes, Isra. No, yeah, Isra. If it's beta, alpha, so the oxygen of the ring and the hydroxyl group of carbon number 1 so far. Just I would like you to remind yourself, okay, the OH of beta structure so close to the O, right? Beta, the OH above the level of the ring so close to O. If the OH below or away from that oxygen of the ring, it's alpha. You got it now, Isra? Okay? Okay.
I got it.
Okay, so just BB has 2 bulbs here and 2 oxygen beside each other. Just remind yourself in that if you will keep on forgetting. Is it hard now? Okay, so this is the glucose structure or the ring structure of glucose. So if I asked you, what is the difference between alpha glucose and beta glucose? Who can answer? Could you write the answer in the chat in words? Sometimes it will not give you structure and ask you, what is the structure between the difference between alpha glucose and beta glucose? Who can say it oral or write in the chat if you know, or you can say oral. Who can say it oral? Raise your hand if you can say it oral. Beta glucose, when the hydroxyl group of carbon number 1, not bottom, not top, we will use these 2 words, okay? Below the level of the ring, okay? Right, we will use these 2 words, okay? Below the level of the ring or above the level of the ring, not bottom, not top. Be specific in your wording, below or above. If it's below, it will be alpha. If it's above, it will be beta. Is it clear? Okay, correct. Thank you. Type. About glucose. Okay, We know that glucose, okay, if you know, the glucose is one of the sugars or saccharide which has reducing power. You know that or not? What is the meaning of reducing power mean? It can lose electrons, right, to reduce other structures. You know that or not? Okay. And there is one test we used to do for the reducing sugar when we were in the IGCSE. You remember that? You remember what is the test of glucose as reducing sugar? Huh? طب you know what is the meaning of reducing? Reducing its ability to reduce other compounds, right? It loses electrons to reduce other compounds. Okay? Right. As a reducing sugar, right? It could be tested by, yes, no Islam. It's a test of reducing sugar. It will be by Benedict reagent, right? So test for reducing sugar, it's by Benedict reagent. So test, okay, for reducing sugars, okay, is made or done by reagent called Benedict reagent, right? Okay. Do you remember Benedict reagent or not? Huh? We had it in IGCSE if you had it before. Okay, I will tell you what is Benedict reagent and I will show you how to do the test now. So the test for reducing sugar, it's made by, okay, reagent called Benedict or Benedict test. Okay, what is Benedict? It is the solution made up of copper oxide, right, which can be reduced to copper +1. This one has blue color. This one has brick red color or brown reddish color. Okay. So the idea of Benedict test, I will put that reagent with glucose, right? Glucose will react with that reagent after heating, right? And if the blue color it change into red, it means there is glucose. If the blue color will not be changed into red, it means there is no glucose. This type of test called qualitative tests. We should know the type of the biological molecule which found in a food sample. For example, if I give you sample of food like bread, I will give you piece of bread or apple or any other food like milk. I would like to know if the bread or apple or milk have glucose or not. Do they have glucose? I don't know. Maybe not, maybe yes. So we can make chemical tests, okay, to see if there is glucose or not. The chemical test is called Benedict test. Why it's called Benedict test? Because we are using Benedict reagent. Benedict reagent made up of some compound which contain copper ion, which can give a change in color if it will be reacted with glucose, right? How can I make that test? Okay, let me show you the video, right? Okay, to see how the test will be made, then I will discuss it with you, okay? What she did here, right? Okay, simply she had sample of sugar here. Here is a sample of the sugar in that test tube. Okay, this is sugar solution. I can use sugar solution, or you can use apple juice, I can use milk, any food sample that you have, right? And this one, which is blue, is Benedict reagent, right? We will buy it for qualitative test. She will take few drops, or not few drops, it's supposed to be equal sample of the sugar. Yani if the sugar 2 centimeter cube, she should add as well 2 centimeter cube or more. I will give you the steps now how to do, right? And next step, we will heat the mixture together. Mixture of glucose and Benedict, we will heat them together. Okay, let's see the test together.
Glucose, maltose and fructose are called reducing sugars. To test for them, add a few drops of Benedict's reagent and heat gently. Sugar changes the colour from blue through green to a yellow-orange or red. The nearer the final colour is to red, the more reducing sugar is present. So
did you see what happened? The color of Benedict was blue, right? It was blue. When you start to heat in boiling water bath, must be at temperature from 80 to 100, right? The color will change from blue, okay? It will start to be gradually green color, okay? Before that, it was green. See, here it was green. Then it will go to yellow, okay? Then it will go to orange, okay? See, it's changing from yellow to orange, okay? If it will keep on heating, it may go to red according to the concentration. And if there are a lot of glucose here, it's supposed to be brick red color. You got it?
So which of these contain reducing sugars? Bread in tube 1, nuts in tube 2 and grape juice in tube 3.
Okay, now she will try with different food sample. First one, it was sugar solution. Yani she took some glucose, dissolved it in some water, and she did it in glucose solution. Now we have different food sample, right? And let's see it will give the same result with Benedict or not, okay?
Bread and grape juice test positive. The nuts give a mauve colour, not a positive result for a reducing sugar.
So according to that, this one give yellow. Yani it changes from blue to yellow or green or red or orange. All of this color considered positive for Benedict. So this one contains reducing sugar. This one as well contains reducing sugar. This one, no, it gives purple. And we never said that the food will give with Benedict purple color. You saw the change. It was gradual from green to yellow to orange to red. You got it? Okay. If I will give, or I'll make Benedict solution, Benedict test in the lab, okay? This is the color of Benedict if there is no sugar. If there are few amount of sugar, or it will start to change in color, okay, it will give that green. So it's written here, traces of reducing sugar. More reducing sugar, as we saw in the video, it will stop at orange. But if it's very large amount of reducing sugar, it will give brick red color. Okay, so if I will put Benedict reagent to the glucose and I heated it up in boiling water bath and give me any of these colors, added to them the yellow one, it means positive. It means there is reducing sugar, which is glucose. If there is any color change, not yellow, green, orange, red, it means no reducing sugar. But how can I do that test? Yani if they will ask you, how can I do the test of the reducing sugars in the lab? How can I do that? Okay, let me show you the steps and how we should write the Benedict test if they ask it how to test the presence of reducing sugar. If we have solid food sample, if I had solid food, okay, sample, we have to crush it first, okay, using mortar and pestle. If it's solid, okay, then add water okay, to the crushed food. Then take 2 centimeter cube of the food, water, or food extract, whatever yani. You can take, sometimes if tomato or grapes, we can squeeze it to take 2 centimeter cube. If it's liquid directly, I will take 2 centimeter cube of the liquid. Okay, so next step, add 2 centimeter cube of Benedict reagent, right, to the food sample. So pay attention here. We have to add equal amount of the liquids from the food and from the reagent. Next step, heat the mixture in boiling water bath. And it must be boiling from 80 to 100 degrees Celsius. Below than that, results will not be given. Okay? What will be the result? If it's positive result, the color will change from blue to green. It's gradually change, even if it has high concentration, to yellow, as we saw in the video. Then we'll go to orange, right? Then we'll go to red, red color. Okay, this is the gradual change of Benedict result. طب, the change could stop at particular color. Yes, it will stop at particular color of this change depending on the concentration of glucose. Yani, the color change could also indicate the concentration of glucose. From here to here, here is no sugar, as we said. Here is high concentration of glucose. Is it clear? Okay. Yes, there is. Yes, Hans. You have a lot about that part. And from here to here, more in traces, as we saw. Right? Here is more and more. Here is the highest concentration of glucose. You got it? Okay. So this is the test of reducing sugar. Okay. Is it hard? Type, can I do that test to compare the amount of reducing sugar between two samples? Type, let me show you an experiment and you can ask, I will ask you to tell me. I have here, okay, apple juice, and I will put orange juice. Okay, apple juice and orange juice, I have two samples of food. Okay, in that one I did Benedict reagent. Okay, and the result here was brick red color. This was the result. Okay, which is brick red color. I did the same test with the same amount of orange, right? And the change stopped at orange color. Or let's make it green. So which one of them has high concentration? Apple or orange juice? Which one of them has more, okay, glucose? Which juice, okay, has more glucose? Here, the result, okay, stopped at orange color. Could you write in the chat? Excellent, Hamza. Apple. Why? Because the net result, okay, will be red, which has the highest concentration of reduced sugar. But orange, no. This sample of orange juice have not a lot of glucose like the apple. You got it? You understand? And of course, to do that comparative test, we must use the same amount of juice in apple and orange. We have to use the same amount of Benedict. We have to use the same temperature of water bath to be fair comparison. Otherwise, my result will not be reliable. Then, you understand that part? This for the monosaccharide glucose. Okay? Fine. There is other monosaccharide. We have to learn about it. which is fructose. I'm explaining biological molecule. I was talking about glucose. I finished glucose structure, alpha and beta, and now going to the fructose. Okay, now I will talk about the fructose structure. Okay, this is fructose. Fructose is monosaccharide, yes, like glucose. And fructose also hexose. You understand what is the meaning of the word hexose now? Hexose means carbohydrate made up of 6 carbon, exactly like glucose. So it's made, okay, of 6 carbons. Actually, fructose is an isomer of glucose. An isomer of glucose. What does the word isomer mean? If I will ask you to write the molecular formula of fructose, it's C6H12O6. Exactly as glucose, as I wrote here, right? C6H12O6. Okay, so why they are different? Because their structure is different. Yani have the same number of carbon, hydrogen, oxygen, but their structure is different. Shall I show you the structure of the fructose? Fructose has 5-membered ring, not 6. It has that structure. Here is CH2OH. Here is CH2OH. Here is OH. And here is OH. That's it. So the difference between fructose and glucose, the structure, but they have the same number of carbon, same number of hydrogen. Okay, usually I exactly like a man which has head and 2 arms, and each arm CH2H, CH2H. This is the structure of fructose. This is the structure of fructose. Directly you will go to the ring structure of fructose. Is it hard? You got it? You know the difference between the fructose and glucose? And fructose not reducing sugar. I cannot do Benedict reagent with fructose. It will not give me any result. Is it clear? Right? Okay. Fructose is another monosaccharide. Monosaccharide is made of 1 sugar. It's hexose. Hex means 6. So made up of 6 carbon. It's isomer for glucose. Yes, it has the same number of carbon and hydrogen and oxygen, but different structure than glucose. Yani the meaning of isomer means what? Have same molecular formula or number of carbon and oxygen and hydrogen, but different structure. How come? Let's look at the structure of glucose. First of all, it's pentagonal ring, not hexagon. 1, 2, 3, 4, 5 ring, and the same oxygen in the ring, right? Carbon number 1, carbon number 2, 3, 4, 5, 6. Okay, carbon number 2 and carbon number 5, they have like 2 arms, CH2OH, CH2OH. One more difference between that fructose and glucose, it is not reducing sugar. Yani it will not react with Benedict. Okay, not reducing sugar, yani give no results with Benedict's test. You understand now, Hamza? Okay, okay. Type. This only I want you to know the difference between the fructose and glucose structure. That's it. Okay. One more thing I would like to give it to you today, and this will be the end of our session, right? I will talk about the disaccharides. Okay, let's go and speak about di. But we said di means what? Di means 2. Saccharides mean sugar. So they are the molecules which have 2 sugars bonded together. Okay. I will only explain one saccharide, one disaccharide, which is maltose, because I want you to understand two different reactions usually we are handling in the biological molecule. As we said before, maltose made, okay, of two sugars, which are glucose plus glucose. Okay. Which two type of glucose? When I say glucose, you have to ask me, miss, which glucose? We said two glucose we have, alpha or beta. Okay. From now, I would like to tell you all of the biological molecules that we study in our syllabus made up of alpha glucose. Made up of what? Alpha glucose, except only one, which made up of beta, which is the cellulose. So maltose made up of alpha glucose, glycogen made up of alpha glucose, starch made up of alpha glucose. Only cellulose will be made of beta glucose. Is it clear? So how two alpha glucose will be bonded together? To understand that one, okay, let's try to put the, yes, Shahd, which part you want me to repeat? I will say it again. Maltose, as I said at the beginning of the session, we have carbohydrates, we call them disaccharides, which made up of two sugar. One of these two sugar, okay, right? Okay, Shah, let me finish maltose, then I will go back to fructose one more time. So when we're talking about two sugars bonded together, like maltose, I would like to show you how two sugars will be bonded to each other. Alpha glucose and alpha glucose, how they will be bonded to each other. To make it easier for me, right, I will, okay, draw two alpha glucose beside each other. So here I will draw one alpha glucose. Here is the hexagonal ring. The most important one for me, carbon number 1, the hydroxyl group is below the level of the ring. Here is CH2OH. Here is carbon number 2, carbon number 3 here, carbon number 4 is again blue. Obviously, is the opposite and inside the ring. All of the other are blue the ring. Tamam? And here, this one also alpha. This is carbon number 2, carbon number 3, carbon number 4 is down. Here, the CH2OH. Here's carbon number 1, 2, carbon 3, carbon 4, carbon 5, carbon 6, 1, 2, 3, 4, 5, 6. Okay. Right, let's think together which carbon or hydroxyl group can react together. You can see carbon number 1 and carbon number 4, right? Because if I will take that alpha glucose and put it close to the other one, which two carbon will react now so close to each other? Carbon number 1 and carbon number 4, right? Okay, so here the reaction okay, will be between carbon number 1 and carbon number 4. Okay, the reaction which will happen here is condensation reaction. What does the word condensation mean? Simply, we will remove water molecule between the two sugar and took the water out, right? HOH, this is water, right? So the two, what is the leftover? Oxygen, which will link the two sugars together. You got it? So here's the bond now between them is here, between carbon number 1, carbon number 4, and this link called glycosidic linkage. You understand? Again, I will repeat it one more time. So now I eliminated water molecule. Did you see it? Okay. And this bond is called glycosidic linkage. Okay. So after elimination of the water, okay, the maltose will have that structure. And this bond called what? This bond is called glycosidic linkage. It's alpha, it's between two alpha sugar, 1, 4 glycosidic linkage or link. Okay, and the other hydroxyl group will stay as it is without any change. Is it clear for all of you? Now, what is the type of that reaction at which we are linking two large molecules by removal of a small molecule? This reaction is called condensation reaction. Yes, good Islam. This reaction is called condensation reaction. And the one who take chemistry and IGCSE could be familiar with that one. What is the meaning of condensation reaction? It's reaction that linking two molecules together by removal of small molecule like water. So the product of that reaction will be maltose plus water. So this how the two alpha glucose will be bonded together by removal of one water molecule, right, to form that glycosidic linkage, which is called 1,4 because the bonding between carbon number 1 and carbon number 4. This is carbon number 1, carbon number 4. Here is carbon 4, here is carbon number 1. Okay, you understand? This is called condensation reaction, and this is the water molecule which is eliminated from here to give the 1,4 glycosidic linkage. Okay, Hamza? Okay, you have any doubt about that? Before finishing the session, we have, I forgot, I would like to add one more thing for you. The opposite of the condensation reaction, hydrolysis reaction. The opposite of condensation, hydrolysis. When I say hydro, it means water. When I say lysis, it means breakdown. In other words, if I will take that maltose, I will add water to that maltose and heat up, it will go back to 2 alpha glucose. So hydrolysis means break down the molecules by adding water. Is it clear? Everything in the session was clear? Okay. Before leaving, we have to test ourselves, right? Let's go through some past paper questions to see if we'll be able to solve the questions or not. Okay, I think, what is that? It is, first of all, it's a disaccharide, okay, or monosaccharide. I will see the glycosidic linkage. It will be what? Disaccharide. Is it maltose or not? Huh? Is that sugar is maltose? Say yes or no. Yes, Tasneem, you are correct. First of all, it is not maltose. Why it's not maltose? Because we said, Yasha, maltose is made up of 2 alpha glucose sugar. Type this one is alpha glucose, correct. What is that glucose? This is what? 5-member drink. 1, 2, 3, 4, 5. So it's what? This one is fructose, right? So the disaccharide, which is made up of glucose and fructose, it's sucrose, and he's written here in front of you in the question. Sucrose is disaccharide, which is made up of glucose and fructose. Why sucrose is classified as carbohydrate? So here I have to define the word carbohydrate, right or no? What are carbohydrates? They are compound hydrocarbonate chain, right? Which made of polyhydroxyl compound, or they have many hydroxyl. You got my point? So carbohydrates, they are a chain of molecules, right? Which have polyhydroxyl groups bonded to it. You have any doubt about that? Or it also has the formula CH2O. That's why it's called carbohydrate, right? It has the formula CH2O. Hydrocarbonate chain, which have polyhydroxyl group, has the formula of CH2O. Sucrose is a sweet testing, right? Leave that one to Putin. We did not finish that part. About that, describe a biochemical test we can use to show if the reducing sugar were found in the first stage of process or not. I want to see if there is here reducing sugar or not. What I should do is this question carries 2 marks. What I should do, I will do. What is the reagent that I should use? Who can write to me in the chat? What is the reagent that I should use to test the present? Excellent. Good, Hamza. Yes, yes, Islam. Good. Benedict reagent. Right? But 1 mark will be carried for Benedict reagent, and the other 2 marks you will say. We add Benedict test, add Benedict reagent, and heat up. If the blue color changed into green, yellow, orange, red, so there is reducing sugar. If there is no change, there is no reducing sugar. Okay, I think that's enough for today. Okay, thanks for all of you for attending. I hope it was